This is an overview of an article entitled The Association of Genetic Instrumental Variables for Lung Function on Coronary Artery Disease Risk, a two-sample Mendelian randomization study, published in the April 2018 issue of Circulation, Genomic, and Precision Medicine. The relationship between reduced pulmonary function and increased incidence of cardiovascular disease has long been known from observational studies. Lung function is measured by forced expiratory volume, or FEV, and forced vital capacity, or FVC. FEV1, which is one of the measures used in the paper under discussion, refers to the amount of air a person can exhale during the first breath. This measure is complemented by FVC, or the total amount of air exhaled during the pulmonary function test. In this graph, you can appreciate the likelihood of survival without a major cardiac event decreasing across the quartiles of FVC, with Q1 representing the highest lung capacity and Q4 the lowest. ISP plus and minus represent the presence or absence of inflammatory proteins, hinting at the underlying physiological mechanisms. The presented estimates of relative risk are adjusted for several potential confounders, for example, smoking, and underscore the robust relationship between pulmonary function and the risk of cardiac events. Similarly robust associations with pulmonary function have also been observed for incident stroke. This particular figure is from the Jackson Heart Study. It shows the complexity of the underlying associations as well as the importance of confounding variables, many of which might not be measured in a given study. On balance, it's difficult to infer whether observational associations between lung function and cardiovascular outcomes are causal effects or merely reflect spurious correlations, for example, due to confounding or reverse causation. In the absence of an ethical possibility of disentangling these associations in a randomized trial, one can use an instrumental variable method called Mendelian randomization to strengthen causal inference. Mendelian randomization treats genotypes, which are randomly allocated at conception, as proxy variables for the given phenotype, in this case, lung function. Then, instead of simply testing the association between lung function and cardiovascular disease, and worrying about unmeasured confounders and reverse causation, one can test the association between the genetic proxy of lung function and the outcome of interest, and that would yield a more reliable estimate of causal effect. Specifically, it eliminates the possibility of reverse causality, since the genotype assignment always precedes disease onset, and also takes care of residual confounding via random assignment to a particular genotype. However, the causal estimates derived using the Mendelian randomization method are only valid if three critical assumptions are satisfied. First, each genetic variant that one is planning to use as an exposure, usually a single nucleotide polymorphism or SNP, must be robustly associated with the quantity it's supposed to represent. In the case of the paper under discussion, FEV1 or FVC. Second, the genetic variable cannot be directly associated with any potential confounders of the relationship between the exposure and the outcome, which include unmeasured confounders. Note in this figure that confounders have to contribute to the outcome, but they can be spuriously associated with exposure and still bias the resulting estimate. Finally, the genetic proxy variable should not be associated with the coronary artery disease or CAD outcome other than through its effect on lung function. That third assumption, in other words, precludes any pleiotropic effects of the chosen SNPs. In the paper under discussion, the authors employed the Mendelian randomization method to answer the following question: does impaired lung function, as measured by common pulmonary function tests, caused coronary artery disease in a large population-based cohort. As the title of the paper states, the authors used a two-sample approach. That means that the association between potential genetic instruments and lung function was tested in one sample, but the causal association between genetic proxies for lung function and coronary artery disease risk was tested in a different sample. Both samples were large and recent genetic studies of lung disease and heart disease, respectively, totaling more than 200,000 individuals. In Mendelian randomization, the genetic instrument can be one SNP or a score composed of multiple variants. In the study, the authors chose 16 independent SNPs for FEV1 and 10 SNPs for FVC. In the main analysis, the authors found statistically significant protective effects of the genetic proxies for FEV1 when using all 16 SNPs. Note here that the wider confidence interval for the FEV1 analysis when only 9 SNPs were used likely reflect the lower amount of trait variance explained by fewer genetic proxies. The associations with FVC were not statistically significant, although the odds ratios were also less than 1. From these data, one sees that increased FEV1 appears to causally protect against CAD risk, although one cannot infer the same about FVC. Now, one needs to assess whether the assumptions needed for these estimates to be valid were fulfilled in this analysis. The first assumption simply states that the proxy genetic variable must be associated with the exposure. The authors showed extremely robust F statistics. Usually, anything over 10 is considered a strong association, although the amount of variance explained was modest, which could mean that the presented estimates of lung function on CAD risk were overly conservative. The second assumption, that the genetic proxy variable isn't associated with any measured or unmeasured confounders of the relationship between lung function and CAD risk, is by definition impossible to fully test. However, because the genotype is assigned randomly at conception, this randomization is likely to equally distribute such confounders across genotypic strata. One notable exception is confounding by population stratification, or ancestry, but the most recent reports indicate this is unlikely to be a major threat to the validity of Mendelian randomization studies. The third assumption concerns pleiotropic effects, and this is where one must distinguish between two types of pleiotropy. Horizontal pleiotropy, which is what one is trying to avoid here, refers to multiple independent effects of a variant. Vertical pleiotropy merely refers to multiple effects that are downstream of the main mechanism of action, which is not a cause for concern. The authors tested for horizontal pleiotropy using the widely accepted Egger method, which is identical to their main analysis, except the intercept is allowed to be different from 0. If the intercept is found to be different from 0, then the Egger estimate will differ from the main analysis, and that is indicative of unbalanced horizontal pleiotropy. What we see here is that although all the p-values for the intercept are insignificant, the estimates of effect are quite different for FVC across the two methods, particularly the one highlighted with the red oval, which precludes a confident assertion that horizontal pleiotropy is not an issue. The third analysis presented in this table, at the far right, shows results from the weighted median method, which relaxes the assumption of no pleiotropy, and you can see that even though no estimates were statistically significant, the magnitudes remain similar to the original analysis. An additional test for pleiotropy that the authors performed was removing the SNPs that were known to be associated with smoking and height, and that did not considerably affect the results. In conclusion, This study presents evidence that better lung function, as ascertained by FEV1, might be protective of coronary artery disease incidence. On the other hand, the estimates of causal effects of FVC on coronary artery disease were null, and horizontal pleiotropy could not be confidently ruled out. Finally, the importance of satisfying the assumptions behind the Mendelian randomization method cannot be overstated, and the obtained estimates of causal effect are best interpreted with caution, especially in light of the weak instruments used in this study.